Good evening, everyone, and welcome to our Kaplan MCAT Prep for the Win event for this evening. We're going to have molecular genetics on the lineup, and we're super excited to have each and every one of you here joining us tonight. We've got a lot of fun stuff planned. You'll see in the chat a couple of links which I wanted to alert you to here in a second. But first and foremost, let me introduce to you who's here. So we've got Brandon and Zinga, who I'll introduce to you in person later. Patrick, Timothy, and Carrie all joining us here tonight, all veteran MCAT instructors. My name is Petros. I, too, am a veteran MCAT instructor and have the pleasure of working with these individuals on a regular basis. And tonight we have the pleasure of working with each and every one of you. So a couple things for you to note. Number one, if you want to find out more information about kind of what's the right steps for you for your MCAT preparation, we do have a link there that you can actually schedule time to speak to one of our MCAT instructors. That's link number one. This is at the top of the chat. But even more fun, we will be giving away a $50 gift card to Target at the end of tonight's event, because you can't have prep for the win without the win. So the win is going to be a $50 gift card. All you have to do is fill out that form there, and you'll be entered into the drawing. Ashwarya, who is also here joining us here tonight, will be selecting the winner, and we'll be sharing that at the end of the evening and the end of the session. So with that said, let me kind of walk you through what we're going to do tonight. We're going to do a quick overview of the MCAT, and then you're going to have a chance to see and actually participate in a MCAT channel session. The MCAT channel is an exclusive feature of Kaplan's MCAT programs. It actually delivers about 100 different individual sessions in addition to the sessions that you have as part of your course that are taught by our top faculty from throughout the country. So everybody gets access to our top teaching faculty. Everybody whose name I mentioned here tonight is a member of the MCAT channel, so somebody that you will interact with in the future at some point when you are taking your CAP course and getting ready for the MCAT. At the same time, when we're doing the channel session here, of course, tonight, you're going to actually see Nzinga, who's one of our MCAT channel faculty members, who's an on-camera teacher. She's going to be the person who's going to be teaching that class, and I'll introduce you to her a little bit later on. With that said, we'll take that as we go and learn from Nzinga about all this fun stuff about molecular genetics. At the end, we are going to talk about kind of what it is that you need to be doing as far as your preparation. So literally, you've got a group of MCAT veteran faculty members here who we're going to be ready to answer all your questions about kind of what you should be doing and when you should be doing it. So we call that plus coaching, but really what that is your opportunity to ask questions and get them answered, and we're gonna do that for you here tonight. So we're really excited about this, and then that's when we're also going to give away the $50 gift card. So lots and lots of fun. For those of you that were here last night, this might be a little bit redundant, but just to make sure that we're all on the same page, the MCAT as an exam is a 7-hour exam, and something that you have to start thinking about right from the get-go is that while you have all these different subjects that you're responsible for on the MCAT, really what matters is knowing the content and knowing how to apply the content, and knowing how to apply the content in the amount of time that is given to you. And that is what our program does. We teach you content, we review the content that you know, but then we teach you how to apply it, and we teach you how to apply it in the amount of time that the test is giving you. Now, the MCAT as an exam is a little different from exams that you've taken as undergrads or that you've taken in school, in that it actually integrates the different sciences together. So you have lots of different pieces that are all coming together in one place. And so that's why we have these 4 different sections, the chemical and physical foundations of biological systems, the biochem foundations of living systems, critical analysis and reasoning skills, which actually has no science on it, and then psychology, social and biological behavior, biological foundations of behavior. These 4 sections each have an average score of around 125. The average score on the MCAT is around 500. That means that's the 50th percentile. Typically, though, you'll see students who are admitted to medical school roughly around the 85th percentile. So the numbers are important. But as I say to all my students, I'll say this to you: don't fixate on the numbers. Fixate on learning how to get more questions correct. That's what we're going to be talking about here tonight, and that's what you're going to experience in this channel episode, is actually seeing kind of what it is that you could do to get more questions correct along the way. I alluded to this a couple seconds ago as far as the integrated nature of this exam. This is a nice little chart. It's available on our website, but also something that's available for you to look at right now. is how the test is actually testing on all these different sciences. And in fact, biochemistry, that's the light blue that's there, you'll see that that actually shows up in equal parts in both the biological and biochemical foundation section, as well as the chemical and physical foundation section. So it really has these two equal parts, or two sections that test it equally. And that's because you can't really separate out the sciences, especially when you're looking at the human body, right? So end of the day, something like the circulatory system might be a biological system, but in fact, it is really fluid dynamics, right? It's a series of pipes with a pump, and the fluid has to go from the pump and get itself back to the pump, right? Circulating through the body. And so as you think about the sciences, you have to start thinking about the integration of the sciences. And as you start thinking about your preparation and your getting ready for this exam, it really needs to be all about making sure that you are thinking about all those pieces and really training yourself to be successful across the board. It's not just good enough to know stuff, you have to know how to apply it. And that's what we teach, is we teach you the stuff and we teach you how to apply it. And that's what you're going to see here in tonight's session. At the same time, and this is just to kind of set you up for what you're about to see, the MCAT also tests you on very, very specific science skills that are outside of content. So general science knowledge is, of course, important, but also scientific reasoning, problem solving, experimental design, research design, statistical analysis, and data analysis, all areas that are important to build your MCAT expertise, and also just knowing how the test is going to test you, knowing what the typical wrong answer choices are, what the typical traps are that are built into the exam and built into the answer choices. So really, really important to kind of think about these things and to look through these things and to learn them accordingly. So you're going to see some of this here tonight as we go through our channel session. And speaking of our channel session, what we're going to be doing is you're going to get a full MCAT channel session today. So we're going to look at molecular genetics. The really, really awesome thing is you can ask us questions throughout the entire viewing. So if you're in full screen, you're going to need to toggle in order to use the chat. But bottom line, you will be able to ask your questions, and we are going to answer them for you. That's why we've got a crew here of more people than I can name right now that are here to answer all your questions throughout this evening. So I'll be here as well. But the person who's going to be teaching you is Nzinga Mack. And Nzinga Mack is one of our, gosh, I want to say, Nzinga, we've known each other for maybe at least 7 or 8 years that we've known each other. We've worked directly with each other. I can think back to a conversation, or first conversation with you probably back in 2010 or so. And you've been here longer than that. Nzinga is one of our rock star MCAT instructors. She's a couple fun facts that she said that I should share with you, and maybe you'll talk to her about this as well. She's a tennis fan, and her hair is 5 feet long, so that's kind of awesome. So we're going to see Nzinga in action. Nzinga, as I said, she's one of our channel faculty members. She is very well loved by all our students, and really the thing that always impresses me getting to work with Nzinga is her commitment to the success of her students, and I think you're going to see a lot of that here tonight in our little preview. So with that said, and without further ado, I'm going to invite Nzinga to come on camera. When Nzinga is done, I'll be back, and we'll kind of talk about the class, but we'll also talk about figuring out what your right steps need to be in order to get yourself set up for success on this exam. So Nzinga, come on cam, and I'll see you all here in about an hour or so.
Well, hello, hello, hello. We're definitely very excited to have you here with us for today's session. I'm excited to see you, and we're going to be talking about some of the major concepts that you want to make sure that you know about in order to do well on the MCAT for test day. So are you excited? Are you ready to learn or to review some of these concepts so that you can make sure that you can knock these questions out of the box when it comes to test day? I definitely hope that you are, in fact, are. Okay, so good deal. So let's take a look at what we're going to be dealing with in today's session. We are going to be taking a look at a wide variety of different topics. So specifically, we're going to be taking a look at replication and what you need to know about replication. And then we are going to be taking a little bit of a look at transcription. We're going to be taking a look at some operons, some base repair mechanisms that you need to know about for test day so that you can be ready to rock and roll on these particular questions. So I see that you're ready, so I'm ready too. Let's get to it, okay? So let's take a look at the first thing that we're going to talk about, the first thing that we're going to review. We are going to review DNA replication. So when it comes to DNA replication, we have some amazing things that go on inside our bodies every single moment of the day. So we have like 3 billion base pairs. So you're talking about this is constantly happening, and you know that you can fit about 10,000 of your cells, if we average out the size of all of our cells, on the head of a pin. Isn't that absolutely insane that you can fit 10,000 of these cells on the head of a pin? That's crazy when you think about all of these things that are going to be happening in these really, 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 really tiny cells that we have. So let's kind of take a look at some of the basics of the mechanism that you need to make sure that you know about for test day. We're going to be taking a look at some of the enzymes that you need to know, but before we get to the enzymes, we're going to be thinking about, well, we definitely do know that we have these 2 strands of DNA, and we know that they're going to run in opposite directions. Can you remind me, what's the name that we call it, indicating that these 2 strands are going to be running in opposite directions to one another? So what is that name that we call it? So we know that we're thinking about the idea of it being anti-parallel. So we know that one strand is going to be going in the 5' to 3' direction, and we know that the other strand, that the matching strand, is going to be going in the 3' to the 5' direction. Good. Now that's going to be important because we're talking about where those sugar phosphate backbones are going to be attached on the carbons of the sugar, okay? So make sure that you note that. If you ever wondered what the 5' and the 3' was going to be about, that's exactly what it's going to be about. So we definitely do know that we have our sugars, we have our nucleotides, we have our bases, we are going to have our phosphates. So we have our sugars, our phosphates, and our bases that are going to make up the DNA. So on one strand, we have those, and on the other strand, we have the matching segments. So as you know, A goes with your, remind me? So we know that your A is always going to go with your T. You know your G is always going to go with your C. You're also expected to understand about the hydrogen bonds that are going to be connecting the bases, okay? So one of the things that we can kind of think about is that you have these very, very strong bonds that are going to be connecting these bases. So it's going to take a lot of work to undo some of these really, really strong bonds. And anytime that we need to take a lot of work to do something, you know that we give that job to an enzyme, okay? So we're going to have some enzymes that are going to unzip the DNA. So can you remind me, what's going to be the name of the enzyme that's going to unzip the DNA? And this enzyme is going to act kind of like a tab that's going to help us pull and tug in order to open that DNA. So we definitely have that awful joke that, you know, if I was an enzyme, I'd be DNA helicase if you're trying to pick somebody up. So I could unzip your, we'll keep it PC, G-E-N-E-S, okay? So we're talking about the idea of your DNA helicase that we have here. Good, okay? So DNA helicase is going to be one of the first enzymes that's going to be active when we're trying to replicate this DNA. And as we know, we have some cells that are going to be replicating all the time. So like epithelial cells, especially cells that are going to be lining your oral cavity, cells that are going to be in that alimentary canal. They're going to be constantly turning over and turning over. As we can also think about skin cells, those are going to be other cells that are going to be constantly turning over and turning over, where the other cells may not turn over as quickly. So for example, our neurons are not going to be turning over all that much, okay? So first thing to do, we know that we need to unzip these G-E-N-E-S, so we're going to use DNA helicase in order to unzip these genes. Okay, so now this DNA is going to be twisted into that double helix conformation that you all know about. So if the DNA is going to be twisted into that double helix conformation, we are going to need something when we open it to prevent it from getting super duper twisted up, to prevent it from getting super duper super coiled, okay? So if any of you remember actually interacting with a phone cord, you know, like the old school phones that were on the wall and you had that phone cord that twisted and twisted and twisted, well, you know that if you untwisted it and opened it at one particular spot, the rest of the cord would get super duper twisted. And you know what? That's exactly what happens to the DNA. So can you tell me what's going to be the name of that enzyme that is going to be responsible for making sure that the rest of the DNA doesn't get super coiled when we're going to be opening one particular segment of the DNA? So if you're saying topoisomerase, then you're absolutely correct. Good. Okay, so that's exactly what we're thinking about. And by the way, I do want you to note that if you're a biology major or if you're a biochemistry major or something to that effect, the MCAT doesn't require all of that knowledge that you had to learn in order to pass your biochem class or in order to pass your cell bio class. We always say that the MCAT is going to be the jack of all trades and the master of none, that it just wants you to get an overview of what that information is. So you may not need to know as many details for the MCAT as you may think that you need to know for the MCAT. So keep that in mind. The MCAT is primarily going to be a critical thinking exam, and that's going to be something that is absolutely necessary to know. Okay, so now let's take a look at another enzyme that you need to know about, and this next enzyme is going to be our enzyme that's going to synthesize some RNA primers. Okay, so can you tell me? And the name kind of goes with it. Okay, what's the name of the enzyme that's going to be responsible for synthesizing some RNA primers in order to indicate to your enzyme, I don't want to say the name, where it's supposed to start laying down the matching base pairs? So there's going to be an enzyme that's responsible for laying down the primers. And like you said, like Lydia and Gregory and Amy and Christy indicated, yeah, we know this is going to be DNA primase that we're going to be looking at. Good. Okay. So DNA primase says, hey, this is where we're going to start this replication. So we know that once the DNA polymerase is going to be able to bind, it's going to know exactly where to, in fact, start. Okay. So DNA polymerase, as we know, is going to be laying down the base pairs. Now, can you remind me, which particular direction does DNA polymerase lay down the base pairs? Does it go from the 5' to the 3' direction, or does it go from the 3' to the 5' direction when it's going to be laying down the matching base pairs that it needs to lay down? Okay, so as you are going to be saying, DNA polymerase is always, 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 like Jonathan and Rachel and Christy and Nicole are saying, yeah, it's always going to be going in the 5' to the 3' direction on that, what's the name of that strand that the DNA polymerase is going to be going from the 5' to the 3' direction on? So we have the two names for the strand. So the one that the DNA polymerase is going to be going in the 5' and 3' direction on, we know that's going to be our leading strand that we have. And we have the other strand, which we know is going to be called the lagging strand. And as we're thinking about that lagging strand, you definitely know that you're going to have a certain type of fragment that is going to be created as we are going to have your DNA polymerase still trying to lay down bases in the 5' to 3' direction. So can you remind me, what's going to be the name of those fragments that you want to make sure that you know about for test day? So it has a special name that you want to make sure that you're thinking about. So we know that these are going to be your Okazaki fragments, okay? So make sure that you remember that concept, because that's going to be something that you need to make sure that you know. And you hear me saying, make sure that you know this concept, because you have to know a lot of different concepts, okay? But you don't have to know a lot of them in great detail. So please make sure that you remember that, okay? So then we want to make sure that we're understanding what enzyme is going to be necessary in order to join those Okazaki fragments, okay? So we're thinking about our DNA ligase that's going to be joining those pieces of DNA that are going to be synthesized on that lagging strand so that we can make sure that we have a continuous strand of our DNA, okay? So as long as you know these particular names of these enzymes, you will absolutely be good as it relates to test day, okay? So let's talk about some other primers that you need to know about. So you're thinking about some of the RNA primers that are going to be super important for replication that you want to think about, that we're going to have about 4 or 5 of these RNA nucleotides that are going to be, like we mentioned before, at the spot where DNA polymerase is going to start its replication, okay? So once we're done with these primers, we definitely do know that we're going to cut out these RNA bases, and we know that our DNA polymerase is going to put the bases down that need to be there. And then what enzyme, if you had to guess, what enzyme is going to be responsible for fixing and connecting the pieces of DNA that were separated when we had the RNA bases there in order to show us where to start, where we could start our replication? If you said ligase in your mind, then you're absolutely correct, okay? So ligase, once again, is going to do this job also of connecting the base pairs where there used to be the RNA base pairs, okay? When we replace them with the DNA. So definitely know that. Now, let me talk about something else here that the MCAT expects you to know. If we take a look at these single-strand binding proteins, I want you to think of them as clamps on the DNA, okay? So we're doing a lot of twisting and untwisting, and we're doing a lot of manipulation of the DNA. And so we don't want it to be moving around too much while we're doing a lot of this manipulation of the DNA. So that's where our single-stranded binding proteins are going to be coming in. They kind of clamp the DNA down so that it doesn't move too much while we're going to be doing all this manipulation of it, okay? So that's going to be the main idea behind our DNA replication and the main process and the main enzymes that you're expected to know about for testing. There are always other enzymes, and if you ask me, hey, Nzinga, what about this enzyme? Or hey, Nzinga, what about that enzyme? I'm going to say yes, it absolutely is there. It is absolutely a part of this process, but what we're focusing on in this class and what we focus on in our channel classes are going to be the most important aspects of the topic that the MCAT wants to make sure that you know about, okay? So think about that idea. So you know what? Let's switch gears a little bit and let's take a look at some adaptations to replications that are specifically in eukaryotes, okay? So I want you to think about this. Let's say that you were trying to copy a book by hand. Let's say that it was a 3,000-page book, okay? And you were trying to copy that book by hand. So would you want one person writing the entire book by hand? Let's say that you had to make, 10 copies of that 3,000-page book. Would you want one person writing that book by hand, 10 times for those 3,000 pages? More than likely, you probably said no, okay? So what could you do instead in order to make that process go a little bit more smoothly that we're thinking about, okay? So we want to make sure that you're thinking about what we have adapted in our eukaryotes in order to make it a little bit easier for this process to happen when we have to copy millions and millions of our bases. So what you would do instead, probably you'd say, you know what? Let's go ahead and have one person copy this first chapter, and then we'll have a second person copy these next 3 chapters, and then maybe we'll have a third person copy this next 5 chapters, and then we're going to put it all together. And with that work, that teamwork that's going to be working at the exact same time, we know that we are going to be able to get through this process more quickly and probably more accurately if you have more people that are going to be doing a little bit less, as opposed to one person who may be super tired by the time that they finish copying all 3,000 pages of this particular book, okay? So that's what we're going to be thinking about. And you said it, Victor, these are going to be the multiple origins of replication that you want to make sure that you know about for test day, okay? So as we look at this, that's exactly what happens, that we have several spots all happening at the exact same time where replication is going to be occurring, and then eventually they're going to join together, and then once they join together, we're going to have that completed piece of your DNA, okay? So then let's also think about another adaptation that we have in eukaryotes, and that's going to be the idea of your telomeres, okay? So if you think about your chromosomes, we definitely do know that we have base pairs that are going to be added to the end, because as we do replication, the RNA polymerase can't lay down the base pairs for the matching base pairs for the very ends of the chromosomes that it's going to be replicating, okay? So since the DNA polymerase, excuse me, can't lay down the matching base pairs at the very end of the chromosome, then we have our telomeres, which are going to consist of repeats of base pairs. So that, you know what, if that particular end gets cut off, it's absolutely okay. It's absolutely fine if that particular end of the chromosome is going to get cut off, okay? And we want to make sure that we can think about how useful this is. Do you know how many rounds of replication approximately can we go through? And by we, I mean like a chromosome. Can we go through before enough of the DNA gets cut off so that it starts to actually affect DNA that's going to be coding for a gene? If you had to guess, what would you say that that is going to be? So it's going to be anywhere from about 50 to 70 rounds of replication that we can go through before we actually hit any of that DNA that's going to be coding for a gene, okay? So definitely make sure that you think about that. And you can also kind of relate this to what you're going to be doing as a physician. You can also relate this to disease, okay? So can you think of a disease in which there's premature aging, advanced aging, and part of the reason why, besides just a defect in one particular protein, but part of the reason why is going to be a defect in telomeres. So what disease can you think of that relates to defects in your telomeres that we have? So you'll note that you can think of progeria, okay? So you may have seen some specials on TV about progeria and, you know, the progerin protein and how it's defective, and it definitely causes advanced ages, but it also has to do with telomeres being defective also, okay? So definitely always a good thing for you to connect what you're learning in class to some of the diseases that you're going to be treating when you actually become a physician, okay? So you know what? Let's take a look at another aspect of replication that you want to know. But before we do that, let me ask you this. Why is it not important for prokaryotes, for our common bacteria, to have telomeres? Can you tell me why? Why would it be not quite as important for them? Where they're going to be like, you know what, this is not a big deal. I don't need telomeres in order to preserve the last base pairs, because they're going to get snipped off every time you do replication. I want you to think about how their DNA is set up, okay? So we know that they have circular DNA, exactly like McKenna and Rachel and Nicole and Katie and Lauren and Layla and Luda and Em. Good, okay? So that's a good thing to note why we're talking about specifically prokaryotic that are going to be adaptations that we have here, okay? So now let's take a look at how we figured out how the DNA replicated itself. So if we're thinking about DNA replication, and we know that this is going to be a semi-conservative process, and by the way, we definitely do know that Watson and Crick, once they discovered the structure of the DNA, we won't mention the fact that they kind of stole Rosalind Franklin's X-ray crystallograph. We won't talk about that, or about how, you know, they were mean to her and they would talk about her behind her back and they stole it out of her lab without her permission. We won't talk about that. But they did postulate, once they took the stolen X-ray crystallograph and once they figured out what the structure of the DNA must be based upon that X-ray crystallograph, they did postulate that, you know what, probably we have some sort of template action going on, okay? So we definitely had to figure out what is going to happen here, okay? So we didn't figure it out for sure, until we went through the Michelson and Stahl experiments, in which they radio-labeled some of the bases and they kind of weighed it to see, okay, where exactly are these bases going to be? So we know the classic semi-conservative idea that, you know, one strand of the DNA is going to be the template and DNA polymerase is going to match that template. And this is what you know, and this is exactly what is going to be happening, okay? But there were some other ideas. So some people thought that maybe there was a conservative method in which you had the old DNA stay together and then you had two completely new strands of DNA that in fact stayed together. And then there was another idea that people had, that maybe we were thinking about a dispersive method. Well, maybe some of the strands of the DNA stayed together for the old one, and then sometimes we had the new one interspersed, and then we had the old one, and sometimes we had the new one interspersed, okay? But until Michelson and Stahl did that experiment, radio-labeling the bases, that's when we figured out that it was going to be semi-conservative, okay? So what I want you to do, we're going to do what we're here to do some questions so that we can make sure that we're ready for any question that we're going to see on test day. Now, there are going to be some questions that you'll be given that won't be associated with a passage. And we at Kaplan, we call these the discrete questions, that they won't be associated with a passage. Or sometimes you'll have questions that are given to you within a passage, but you don't actually need the passage in order to answer the questions. And sometimes, not always, sometimes some of those questions are pretty straightforward, okay? So anytime we see some of these questions being pretty straightforward, we're super excited, we jump up in the air, we click our heels, and we go ahead and answer it, okay? So what I want you to do, we'll pull up the poll, and I want you to take a couple of moments, and I want you to answer this question, okay? We want to know the process of DNA synthesis follows this order. So make sure that you go ahead and we'll give you about maybe 15 or so seconds in order to answer this question. Make sure that you don't put the answer in the chat. Make sure that you put it in the poll only. So this is a question that we don't expect to take a lot of time, and that is absolutely wonderful, okay? So we take a look at this, and it looks like 95% of you indicated that the correct answer choice is C, and you're absolutely correct, okay? So we have to unwind, we have to make sure that we pair the matching base pairs, and then we want to make sure that we're joining the base pairs that in fact do need to be joined, and that we're joining the pieces of DNA that need to be joined. Excellent, okay? We see a question like this, we're super excited because we know that there are going to be some questions that's going to take a long time to do. And by a long time, I mean over a minute, and definitely over a minute and a half is going to be super duper long when it comes to doing any question on your MCAT. So that's a nice thing for you to keep in mind in terms of the amount of time you should be spending per question approximately. But yeah, you just did this in like 15 seconds. So that means that you have all that extra time for another question that may take a little bit longer, or all that extra time to read a passage that may take you a little bit longer to comprehend exactly what's going on. Okay, so good deal. So you know what? Let's go ahead and let's take a look at another topic that you need to make sure that you know about for test day. And that next topic is going to be a question about what we need for DNA replication. So you know what? We're going to give you like another 15 seconds or so. So see if you can go ahead and answer this poll and see if you can come up with the correct answer. So this question is a little bit tricky. We're being asked which is not a requirement for DNA replication. Now on test day, one of the things that you want to note when you're taking a look at the question and assessing the question, and by the way, that's one of the things that we do as part of our Kaplan method, is that we assess the answer choices to see if there are any trends in the answer choices that'll make our lives a little bit easier. We kind of like these answer choices because they're relatively short. So we're fans of short answer choices because that means that we don't have to spend a lot of time reading and understanding what's going on. We can kind of move on to get the question correct. So we see this, and then we see a not in the question stem. So we have to be super duper careful with the nots. Because even though the not, has that ever happened to you? Where there was a not or an except in the question, but you still picked the opposite answer choice anyway? And then afterwards, when you reviewed it, you were like, oh man, why did I pick that? There was a big not or a big except that was going to be right there. And I still picked the opposite answer choice. So we do like a little bit of a check to make sure that we see what's going on. Okay. So we have this, and I want you to note that you will often see a lot of questions that have the answer as none of the above on test day, but every once in a blue moon, which I think actually may be coming up at the end of this month, but every once in a blue moon, you may see a question that has an answer choice that is none of the above. And that is the correct answer choice that we have here, okay? So we think about this and we're like, not a requirement for DNA replication. We're like, man, that's a broad question. Okay, let's see if we can come up with a prediction. We need a whole bunch of enzymes. We need a whole bunch of working parts. We need some ATP. So we look at this and we're like, okay, we need deoxynucleotide triphosphates, okay? These are definitely going to be the bases, okay? So just in case you're wondering what your DNTPs are, those are the bases, our A's, our T's, our C's, our G's. So definitely make sure that you can recognize it in that particular name. Of course, we need some ATP. All of this is a very energetic process. This is not going to happen spontaneously, okay? So since we know that this is not going to happen spontaneously, we need to make it happen. And as we talked about previously, we're definitely going to need a primer, okay, where we have those RNA base pairs that are going to be laid down in order to show the DNA polymerase where it needs to start the replication, okay? So in that case, our correct answer choice is going to be D. That is going to be none of the above that we are going to be looking at here, okay? So good things for you to think about, okay? So awesome, awesome, awesome. I love the fact that you're well-read when it comes to the idea of medicine. That means that you're really, really interested in. And there's some things that are really interesting. There's some things that are like interesting good, and there's some things that are interesting bad. But you know, it's interesting all the less. And I love the idea of that book club, making that book club happen so that we can make sure that you know what to do and that you know what's going on in the world around you, okay? So with that, let's go ahead and let's continue on. And what we're going to be taking a look at, we are going to be looking at some of the ideas that are going to be involving repair of the DNA. So we have some common mechanisms that we want to make sure that you understand, okay? So we definitely do know that the DNA is going to get proofread as it's going to be replicated, and that's going to be key. And the amount of mistakes that DNA polymerase makes is like crazy infinitesimal, that it really makes mistakes. But every once in a while it does. And we want to make sure that we know what happens when it does in fact make some of those mistakes. So when it comes to DNA being messed up, so one of the things that I do, I'm a PhD student and I do cancer research. So I'm researching triple negative breast cancer cells and how they're going to be reacting to the knockdown of the enzyme lactate dehydrogenase. So I've taken some classes in cancer biology, and one of the first things my professor told me is that, you know what, when you die, everybody has a little bit of cancer in them. Everybody has a bunch of cells or several patches of cells all over them that the DNA has in fact gone rogue. And what causes the DNA to go rogue? You tell me. Actually, you tell me. What are going to be some things that can cause some, like your mismatch repairs, or your mismatches in the bases, or where you have this kind of thymine bridge where the two thymines bind to one another instead of binding to adenines and cause that little kink there, or that are going to cause a base to be inserted or a base to be deleted. What are going to be some of the things that cause that? Okay, so we think about your UV light, anything that's a mutagen, okay? And by the way, like everything is like a mutagen, whether it's going to be like the air quality, whether it's going to be the paint on the walls, whether it's going to be the the materials that you use, or the products that you use for your body, your face, your hair, everything, okay, is a carcinogen, like the food that you eat, in terms of if you eat some of the burned food. So the main idea, tanning beds, pesticides. So the main idea is to make sure that you are as protected as you possibly can be, okay, within the realms of protection. So H. pylori, everything. So you definitely want to make sure that you're exercising, that you're eating well, that you're doing everything that you possibly can in order to make sure that your immune system is ready, and in order to make sure that your immune system can tackle any cancer that it sees. We can nip it in the bud right then and there. American food, there you go, John. Okay. So with that, we want to note that, yeah, X-ray, your V, is going to lead to the damaged DNA. So then one of the things that happens is that this process often happens at the same time that we're doing replication, or immediately after we're doing replication. So if you were to ask us, well, what are some of the enzymes that are going to be involved in this? There's going to be a substantial amount of overlap between the enzymes that are going to be involved in this. But we definitely do know that you're going to have your helicase that is going to be responsible for opening up the DNA, or making sure that we can get to the spot that we need to get to in order to fix it, okay? So sometimes you can have DNA polymerase that can sometimes slip. Okay, so you have that whole mechanism where you have the polymerase is going to be attached to the DNA, and sometimes it can slip. And if it slips, it may skip ahead and put a base where it's not supposed to, or it may accidentally get rid of a base or something of the sort. It kind of loses its footing. So sometimes you'll have, more DNA than it's supposed to have, or less DNA than it's supposed to have. So you know that you have a group of proteins that are going to recognize that problem. Sometimes it's going to be a problem that's going to distort the helix, and sometimes it's going to be a problem that isn't necessarily going to distort the helix, okay? So we definitely do know that we're going to have different types of repairs based upon whether it's a problem that distorts the helix or whether it's going to be a problem that is not going to distort the helix. But once we've identified where the problem is, and once we've identified what type of problem that it is, we're going to have a specific type of enzyme that's going to remove the damaged DNA, okay? So what's going to be the name of the enzyme that's going to be responsible for removing that damaged DNA so that we can make sure that we get the problem out? Whether it's your thymine dimers, like Laura is saying, whether it's going to be an additional base pair, or whether it's going to be too little of a base pair, a missing base pair. So we definitely do know that we're going to have your endonuclease that's going to be responsible for removing that damaged DNA. Good, okay? So your endonuclease is going to remove that damaged DNA, and then you're going to have that gap closed, so you can probably guess. So it's going to be the name of the enzyme that is going to be closing that gap. It's going to be the same enzyme because we said it's happening at the same time as your replication. So we know that this is going to be our DNA polymerase that's going to be responsible for sealing up the gap. And then, of course, for making sure that we put the correct base pairs in the gap. And then, of course, we have that ligase that is going to be responsible for sealing up the gap, okay? So one of the things that you want to make sure that you know about, and you can read more about this in our wonderful Kaplan books, is you can go into more detail about your mismatch base repair, your base excision repair, your nucleotide excision repair, and all of those specific types of repairs for the specific types of things that can in fact go wrong with the DNA. And there's something that you know if you've studied cancer, that you have a lot of cancers that take advantage of these particular mechanisms of repair of the DNA. And as they take advantage of the repair of the DNA, they overwhelm these repair mechanisms, and the DNA just kind of goes rogue, okay? So there's a very famous example of a particular gene that is a gene that it is handed down through generations, that if this particular repair gene is damaged, that it's going to lead to breast cancer. So can you remember, there was a very famous actress that actually went ahead and got a mastectomy because she had the gene, her mother had the disease, and she wanted to kind of reduce her chances of getting breast cancer. So good, we're talking about the BRCA1, BRCA2. So these are going to be malformed genes, malformed proteins that are going to be involved in damage repair to the DNA, okay? So definitely make sure that you think about that. Yeah, so we're talking about Angelina Jolie and what she went ahead and did. Good, okay? So always a good thing for you to think about, okay? So you know what? Let's go ahead and let's take a look at another question so that we can see how you can snatch up some quick and easy points on some of these topics when it comes to testing. So go ahead, we'll pull up the poll for you. We'll give you about another 30 seconds or so. So take 30 seconds, put your answer in the poll, and we will review it afterwards. Okay, so let's take a look at what you said for this. I was kind of looking to see what you were saying, what you were doing, and all of those things. So in this particular case, we want to make sure that you know what is going to be the correct answer, okay? So we're trying to figure out DNA replication enzymes can be utilized in DNA excision repair, except, okay, so this is another question where we're going to be talking about the exceptions that you need to make sure that you know about for testing. So if we're going to be thinking about the exceptions, we kind of have to know what all of the enzymes are that we're going to have to use. And you know what? This is another question where you won't see a lot of these on test day, but it's good to know that all of these actually are going to be used like you indicated. So excellent job coming up with that as an answer. Only about 85% of us said that. So you know what? We're going to have to go with the silver stars for that 85% that we had here, okay? But for now, let's make sure that we can turn our attention to another topic that you want to know about for test day, and that is going to be the idea of kind of determining how particular genes are going to be expressed, okay? Whether or not they're going to be always off and we have to turn them on, or whether or not they're going to be always on and we have to turn them off. So you probably know what I'm about to talk about. And specifically, we're going to be talking about it in prokaryotes. And specifically, we are going to be talking about our operons and what you need to make sure that you know about your operons for test day, okay? So we definitely do know that there are other ways in which we can regulate genes being turned on and off. So can you tell me, what's that entire study of ways that we can regulate whether or not a gene is going to be turned off or whether or not it's going to be turned on? Okay, so one of the things that you're thinking about is your epigenetics, okay? So if we're thinking about epigenetics, we're thinking about DNA methylation that's being used to turn genes on, okay? We're thinking about how we can condense the chromosomes a little bit more so that we can make it a little bit harder for a polymerase to get to a particular gene. Or we can loosen up the DNA in order to make it a little bit easier for our replication and for our transcriptional, specifically, processes to in fact happen, okay? So we're thinking about epigenetics. So epigenetics is a fascinating field because one of the things that you can think about is the idea that it's been proven that the way that your grandmother reacted to certain situations can be handed down epigenetically to you, okay? So let's say that your grandmother was in a situation in which she had a lot of stress in her life, okay? So all of the genes for stress are turned on, okay? So if we're thinking about all of the genes for stress being turned on, then that's going to be passed down to the daughter, okay? That's passed down to the daughter. So all the genes for stress are turned on, and then that's passed down to the granddaughter. And it makes sense because I guess nature was like, you know what? If we are going to have all three of these people, if they're going to be in the same environment, then they kind of need to come out into the world prepared to be into this particular environment. So we definitely do know that it can change which particular genes are turned on or turned off over a lifespan. But that's definitely something that's really, really, really interesting to in fact know as it relates to your gene regulation in your eukaryotes, okay? So it's definitely interesting things. But let's turn our attention to gene regulation in our prokaryotes. And we're going to be taking a look at our operons, okay? And specifically, this is going to be a really famous operon that I'm pretty sure that you are familiar with. So this is going to be the lac operon that we're going to have here. So if you take a look at this lac operon, we kind of want to say to ourselves, okay, well, let's see what's going on. So first of all, let's go ahead and let's kind of identify all of the major players that we're going to have here. Okay, so we have the structural portion. So the structural portion of the gene, it literally is going to code for the protein. Okay, so that's going to be something that you want to make sure that you know. The operator is going to be the portion that is going to bind, the portion of the DNA that is going to bind the repressor. We'll talk about what the repressor is in just a moment. Okay, so operator binds the repressor. Okay, and then we know that we have the promoter. As we're going to be taking a look at the promoter, we know that that's going to be where what binds. Can you remind me? The promoter is where what binds. So we know the promoter is going to be where our RNA polymerase is in fact going to bind, okay? And then we have the regulator. The regulator is going to be the portion of the DNA that codes for the repressor, okay? So the regulator codes for the repressor, okay? So now that we kind of have a lay of the land as to what is going on, let's kind of take a look to see what is happening in this particular case. So we have this lac operon. The lac operon, the structural portion of this DNA segment, it actually codes for the protein beta-galactosidase, okay? So this is going to be an enzyme that's going to be used to break down beta-galactose, okay, whenever the organism needs to break down beta-galactose. Okay, so the organism doesn't always need to break down this particular sugar. So since the organism doesn't always need to break down this particular sugar, because it actually prefers glucose when glucose is available, it's like, okay, I want to break down glucose. That's more fun to break down, okay? So this particular gene is going to be in the default position of being turned off. And the way that they turn it off is by binding a repressor protein. The repressor protein that's going to bind to the operator, and it's literally physically going to block RNA polymerase from being able to do transcription, okay? But let's say that there's no glucose and the organism is like, okay, fine, I guess I'm going to go ahead and digest some of this lactose that is around. Then we definitely do know that we are going to have the removal of the repressor protein, okay? And a lot of times what's going to happen is that the repressor protein is going to bind to another molecule. Can you remind me? What's the name of that other molecule that the repressor protein binds to instead of binding to the operator like you see it currently in its default position here? So we definitely do know that we are thinking about your inducer, okay? So we definitely do know that the repressor is going to be binding to the inducer, and typically the inducer tends to be the product that we need to go ahead and make this beta-galactosidase for, okay? So the inducer is going to bind to the repressor comes off the operator, and now we know that our RNA polymerase is free to transcribe this particular gene, okay? So make sure that you understand that process. Now, I think I saw it in the chat. Since transcription of this gene, the default is for it to be off, we call this what type of an operon, since we know that the default is going to be off. So I see some of what you were saying. This is going to be an inducible operon because we can induce it. We can, in fact, turn it back on whenever it is needed according to the environment. Okay, so good things for you to think about there. Okay, so now let's go ahead and let's take a look at another idea that you need to know about. So you probably know what type of operon we're going to talk about next. Okay, we know that we're going to be taking a look at our trip operon. Okay, so as we look at our trip operon, well, we know this is going to be the opposite. So can you tell me what type of operon is the trip operon? So if the lac operon was an inducible operon, we know the trip operon is actually going to be, you got it, Ludia, we definitely do know that this is actually going to be a repressible operon. Okay, so we have the same factors, we have the same major ideas that we have here. The regulator, okay, pop quiz, okay. The structural component of the gene, can you remind me, what is that going to bind for? The structural component of the gene that we have here. So we definitely do know the structural component of the gene is going to bind for the protein itself, okay? We definitely do know that the operator, can you remind me what the operator does? I love it. Victor and Jessica and Ludia and Laura and Darren and Jonathan. Okay, so we know the operator is going to be what's going to bind the repressor. Good. And in this case, the co-repressor. Okay. And then the promoter, we can kind of see from the diagram here, is going to bind your RNA polymerase. And remind me, what does the regulator code for? So we definitely do know that you're going to have the regulator that is going to be coding for the repressor protein. Good. Okay, so excellent. So definitely keep that in your mind as you're going to be looking at what you have here. So you can see here that the default is going to be that the repressor is not going to be added to the operator at all. Okay, it's not bound to the operator, so this gene can in fact be transcribed. And this is actually going to be a gene that's going to help in the synthesis of the amino acid tryptophan. Okay, so this is a prokaryote. It's going to be one of the classic amino acids. And by the way, let me ask you this. Do you need to memorize the amino acids for test day? Do you need to know the structure of all of the fundamental amino acids for test day by heart? Do you have to be able to recognize them? Do you have to know which ones are going to be acidic? Do you have to know which ones are going to be basic? Do you know which ones are going to have rings, aromatic, non-aromatic, etc.? So the answer to that question, luckily or unluckily, depending upon how you look at it, is yes. Okay, you are going to have to know the names, both the full name, the single letter name, the 3-letter abbreviation, and you're going to have to know the structures of all of these amino acids and know whether they're acidic, basic, aromatic, non-aromatic, etc. And one of the things that we always do is we have a series of these wonderful short videos that you get access to once you sign up for our course that give you nice reviews over certain topics that we know our students struggle with a lot. And we have some nice videos that are on amino acids and how to make sure that you know the similarities and the differences between them and how you can help to memorize all of them. Okay, so definitely note that. So we look at this and we're like, yeah, you know, when this is going to be in the default mode, this is going to be coding for a protein that's going to be helping with the synthesis of the tryptophan for this prokaryote. Now, let's say that all of a sudden this bacteria is in an environment where there's tons and tons of tryptophan everywhere. So if there's tons and tons of tryptophan everywhere, then it's not going to be necessary for it to synthesize its own tryptophan, and it can use that energy to do something else, okay, like maybe reproduce or something of the sort. So in that case, we need to shut off this transcription, okay? And that's exactly what we're going to be seeing here, that we're going to have a co-repressor that is going to bind to the repressor. And the co-repressor and the repressor together are going to bind to that operator. And when they bind to that operator, they're physically going to block that RNA polymerase in order to stop our transcription from, in fact, happening. Okay? So good things for you to know. And as we indicated previously, a lot of times that co-repressor is going to be the end product. So a lot of times it's like actually tryptophan that binds to the repressor that is going to bind perfectly to the operator and stop this entire process. Okay, so make sure that you know the similarities and the differences between these two operons that you need to make sure that you know about for testing. Okay, good deal. So with that, let's turn our attention to some of the key players of transcription before we start to talk about transcription itself. So one of the things that you'll note is that we have a little asterisk here, and we indicate that some of these are going to be either prokaryotic or eukaryotic transcription factors, okay? So when we're talking about the first step, when we know that we're going to be talking about the promoter, we want to make sure that we understand that we don't have the Tata box in prokaryotes, okay? So the Tata box, you can find it in archaea, and you can find it in your eukaryotes. But we definitely do know the Tata box is going to be that promoter sequence. So it's going to be a repeat of your T and A and your T and your A bases. That is going to help us figure out where transcription is in fact going to begin, okay? So there's something called a Pribno box that we have in prokaryotes instead, okay? So that's going to be that one. We definitely do have sigma factor, okay? So sigma factor is going to be another one that you want to know about. This is another transcription factor that's going to initiate transcription. RNA polymerase, we definitely do know that RNA polymerase is going to lay down those matching base pairs. But can you remind me, what's going to be a base that we do not find in RNA, but that we do find in DNA? So a base that's not found in RNA, that is not going to be found in your DNA. So we definitely do know that we're going to have our T's in the DNA, but definitely no T's in the RNA. That we know that we're going to have U's in place of the T's in the RNA. Good. Okay, so of course we need to have the ribonucleotides, like we just talked about, that are going to be the base pairs that are going to be laid down. Okay, so initiation, we got to get this process going. Then we have the idea of our elongation. Okay, so we're laying down, continuing to lay down the base pairs. We have our RNA polymerase. We have our helicase. And of course, we know that our helicase is going to be used to continually open up the DNA so that we can make sure that we get that RNA polymerase in there. More ribonucleotides so we can lay it down. Okay, and then you have some termination sequences. So you have hairpin turns that are going to be responsible for being a termination sequence in prokaryotes. That is going to say, okay, we can stop this, because what happens is you're going to have the RNA that goes and goes and goes, and then you have that hairpin sequence that those bases at the very end, they're going to match up with the bases that came right before them. So when they match up with the bases that come right before them, they become a hairpin, and the RNA polymerase can't continue. Okay, we have that rho factor, which is going to be another termination factor or termination component that is going to be found in prokaryotes. Okay, so as one of the things that you can always do, you want to know these particular transcription players, but remember, we always have all of this information available to us in our review notes. So if you ever want to go into more detail about it, you can always go and take a look at it there. Okay, so make sure that you think about that. So then, you know what, let's go ahead and let's take a look at what's going to be happening as it relates to your transcription and what you need to know about your transcription for test day. Okay, so let's take a look at kind of what's going on with the DNA sequence itself. Okay, so as we take a look at what's going on with the DNA sequence itself, one of the things that you can note is that you have a couple of things that go on before we actually get to transcription, okay? So if you're looking at a couple of things that are going on before you actually get to transcription, you see that you have your CAAT box. So can you remind me, what does that CAAT box do? It's a binding site for what? So the CAAT box is going to be a binding site for your RNA transcription factors, okay? It typically is going to be about 70 base pairs away from where we're going to have the origin of transcription, okay? So make sure that you know that about that CAAT box, okay? And then you can see that TATA box. Okay, well, we already talked about the TATA box, and this is going to be the sequence where we're going to have our transcription beginning right afterwards, and that's typically about 25 base pairs, as you can see in this diagram, away from the origin of your transcription, okay? So make sure that you know that. Then you can see where we're going to start transcription. And by the way, we have tons and tons of enzymes and proteins that are going to bind in order to make this transcription happen. So you definitely typically have about 5 to 7 of these proteins that are in fact going to bind. So you definitely have your promoters that are going to contain a bunch of sequence elements in which we can know exactly where the RNA polymerase is in fact going to bind, okay? And we definitely have tons and tons of subunits. So, you know, your RNA polymerase has like a sigma subunit that you want to make sure that you know about for test day so that you can kind of have an idea of what's happening. So, of course, you have tons and tons of RNA polymerases that are going to be responsible for transcribing specific portions of the DNA. But a lot of times, the RNA polymerase that the MCAT wants you to know about is going to be your RNA polymerase number 2, okay, which is going to be responsible for transcribing a lot of the segments of the DNA. So speaking of transcription, let's kind of take a look at the idea behind transcription, that we're going to be making a copy in terms of RNA that is now going to be taken outside of the cell, or rather outside of the nucleus, not necessarily outside of the cell, outside of the nucleus. And we know that we are going to match up that messenger RNA once it's outside of the nucleus, and we're going to match up the amino acids that this messenger RNA is going to be coding for, okay? So we think about this and we're like, okay, that's going to be the main idea that we're going to have here. So you can see here that you have exons that are going to be here. You have introns, Remind me, your exons, are those going to be the portions of the DNA that are going to be expressed? Or are those going to be the portions of the DNA that we're going to cut out when we're thinking about our exons? I'm completely with this joke, Aishwarya, by the way. Okay. So we like this, and we know that the exons are going to be the portions of the DNA that once we make them into the correct heteronuclear RNA, and we'll talk about what that means in just a moment, these are going to be the portions that are going to be expressed. And that's a nice way for you to remember that X is going to be for expressed, whereas the introns are going to be the ones that are going to be cut out. Okay, and we'll talk about what's going to happen a little bit later and what's going to happen with all of the signals that you do in fact have. Okay, so you know what? You tell me, what are going to be some things that after we have decided to terminate this particular transcription, what are going to be some of the things that we're going to have to do to this nascent heteronuclear RNA? And by the way, we call it heteronuclear RNA because it's still inside the nucleus. What's one thing that we're going to have to do before we can have it exit the nucleus? So one of the things, and I like what you're saying, yeah, we have to have a poly A tail, good, okay, we need to have a 5' cap, perfect, okay, poly A tail, and then do the splicing that we did in fact talk about. So I want you to think about the significance of that. I want you to think about, why do we need to have that poly A tail? What is that going to be about? Why do we have to have that 5' cap, you know? So one of the things that we can think about is that we need to identify our own messenger RNA, because every once in a while, and it has been happening a lot during this particular season, other organisms, well, if you want to be snarky, you can say it's not really an organism. But when it lays you flat on your back and has you in your bed for like 2 days straight, feeling tired because you can't do anything, yes, this is personal, then maybe you may have to give a little bit more respect. So what's going to be another organism that can kind of hijack this particular system and make its own messenger RNA and send it out into the cell so that the cell can make more copies of this particular organism? So we definitely do know that we are thinking about like viruses, okay? So we have to make sure that we can identify our own messenger RNA as opposed to a viral messenger RNA, and that's part of the reason, not the complete reason, but that's part of the reason why we have that 5' cap and the poly A tail, where we have all these adenines that are going to be added to the very end of this transcript, okay? And then from there, we send it out to the nucleus, and once we send it out of the nucleus and send it into the cell, then we know that we are going to do what we need to do, and we're going to be taking a look at what's talked about in another MCAT channel session. And the next MCAT channel session is going to be talking about the idea of your translation. What's going to happen to that messenger RNA after we translate it into the actual protein that we are going to be thinking about, okay? So note, can you remind me, how many adenines do we put in that poly A tail? There's definitely like a whole bunch that we put in that poly A tail that we're going to have. So you'll notice it's going to be about 250 or so of your adenine. So it's a tail. It's not like 2 or 3 adenines that you add to the very end of it. It definitely is going to be a major tail that you have here. Now, of course, there are a couple of things that you want to make sure that you know about, because this is not the end of the story. Just because we have an mRNA transcript does not mean that that particular gene is going to be transcribed. We definitely do know that we have enzymes that are going to be responsible for literally cutting up the messenger RNA transcript, okay? They're like, oh, no, we do not want that particular protein anymore, so, you know, let's cut up these mRNA transcripts so that we don't have that protein. Or sometimes you may have proteins or bases that will actually bind to the mRNA transcript, and when these bases are binding to the mRNA transcript, you can't have the translation machinery bind to the mRNA transcript. So that's going to be one of the ways that you are going to have the blockage of the translation, okay? So definitely keep that in mind, that you definitely have several different mechanisms to stop a particular protein from being translated. if we need to stop that particular protein from being translated. And that's actually one of the things that I work on with my research, that I actually use a viral vector in order to stop the lactate dehydrogenase A messenger transcripts from, in fact, being able to turn into lactase dehydrogenase A protein. So it's always going to be very useful to understand what's happening and kind of use the viruses, because they do this very well, in order to do research, or in order to help them do something that we want them to do, as opposed to something that we don't want them to do. And thank you very much for the for the love when it comes to getting over the flu. Okay, so with that, you know what? Let's go ahead and let's take a look at a couple of questions so that we can make sure that you know what to do. Okay, so we'll give you a couple of minutes. Actually, we'll give you a minute for this question. Go ahead and put your answer in the poll and let us know what you think. Okay, so let's see what you're saying for this particular case. I see that we have a bit of controversy for this question. So we're trying to figure out eukaryotic mRNA production is going to occur in what sequence, okay? So one of the things that we do, we take a look at these answer choices, because this is definitely a part of the Kaplan method. And we actually love these answer choices because they're actually pretty similar. So anytime we have answer choices that are really similar, we're like, yes, because what that means is we don't have to do a lot, expend a lot of mental energy figuring out what each answer choice says and then seeing whether or not it makes sense. So we want to see some of the similarities. So like transcription from the DNA, okay, we have in the cytoplasm, in the nucleus, in the ribosome, in the cytoplasm. Okay, so you know what, we can eliminate some answer choices because, you know, they started saying some nonsense right off the bat. So, and by the way, I want you to note what I'm doing, that if you read into an answer choice and the answer choice starts talking about nonsense, you're not even going to finish reading the rest of this answer choice. So we look at this and you see, what are some answer choices that we can eliminate just reading to where I underlined? Transcription from DNA in the cytoplasm, transcription from DNA in the nucleus, transcription from DNA in the ribosome, transcription from DNA in the cytoplasm, okay? So one of the things that you'll note, you take a look at this and you're like, oh, wait just a minute now. Just by taking a look at that, I can get rid of transcription doesn't occur in the cytoplasm. That's not where it's going to go. Transcription isn't going to occur in the ribosome. What? No, it's translation. Okay, transcription isn't going to occur in the cytoplasm. So just like that, you actually get to the correct answer. Okay, A, C, and D are eliminated exactly like Lauren indicated. Good, okay? So we are like, oh, okay. So we know that our correct answer choice is indeed going to be B, okay? It's B, and we like it that way. So I want you to think about this. I don't know if you remember, there was like this really mean dating show. I think it was like on MTV, where you'd have, it was called Next, okay? Where you would have people who were potential daters. There'd be 3 of them on the bus, and the bus would pull up to a person who they were going to go on the date with, okay? So the person outside of the bus that they were going to go on the date with would, you know, be there, and then the first person would come off the bus, and if that person outside didn't like the way that person looked, they'd be like, next, and they wouldn't go on the date, okay? And if that person outside liked the way the person looked, okay, let's go on the date for like 10 minutes, but then the person said something, you know, that they didn't like, they'd be like, next, and then get the second person off the bus. I say all of this to say, this is exactly how I want you to be with these answer choices, okay? If you start going through an answer choice and it says something that is factually incorrect, that answer choice is not going to be the correct answer, okay? So you go transcription from DNA in the cytoplasm, next, and then you move on to the next answer choice in order to make sure that you make the absolute correct decision, okay? Because like I said, and that's one of the great things about the answer choices on the MCAT, especially for the science sections. Even if you have like no idea what the question is asking, if the science is factually incorrect, you can eliminate that answer choice, okay? So remember that idea in order to make sure that you get a lot of points that you need to get on testing, okay? So with that, you know what? We covered a lot in today's session. So we took a look at some of the events involving replication, okay? So can you tell me, what's some of the things that you talked about, that we talked about in today's session about replication? That was a nice review for you, or was something that you were like, oh, I didn't even remember that, or I never learned that. So what's the one thing about replication that you're like, okay, you know, what is going to be going on in this case? So we took a look at some of the ideas that are going to be going with the various enzymes, like your helicase, like your single-stranded binding proteins. We took a look at your topoisomerases, what they do. We had that analogy of, you know, we don't want the DNA to supercoil. We definitely do know that we are thinking about the Okazaki fragments, 5' to 3', 3' to 5', all of those good things. Then we talked about some of the mechanisms of DNA repair. So what's one thing that we said about the enzymes between DNA repair and replication? Okay, so we definitely do know that the enzymes are going to be pretty much the same, okay, that we have between these two, and that basically everything can cause your DNA to, in fact, you know, have something go wrong with it or mutate, okay? We talked about the lac operon, we talked about the trip operon, and what you need to know about it, and then we rounded it up with the ideas that are going to be behind your transcription, okay? So stay tuned. Petros is about to come back on, and what he's going to talk about is super important. Actually, if you ask me, it's actually more important than what we just went over. It's the idea of making sure that you're set up in such a way that you know what to study, how to study, and when to study, so that you can make sure that you do your absolute best on test day, okay? So I hope to see you one day on the MCAT channel. I'll be looking out for you, or maybe in one of my MCAT classes, and maybe you'll be one of my success stories in which the person went from a student of mine to being a doctor and told me about it. That's always something that's very exciting, okay? So I hope to see you in one of my classes so that we can make that happen for you. And thank you, Lenore. Appreciate it. Bye.
All right, so, excuse me, I got a frog in my throat. Big thank you to Nzinga. How'd you guys like that? I know Nzinga was asking you about, you know, kind of things that were on your minds as far as these topics go, and maybe triggered a little bit of a, oh, I need to review that. But kind of in general, you know, it's the good news is that, I think Nzinga mentioned this, the MCAT, very, very preparable exam and very predictable exam. You saw how predictable it was in that last problem there, right? When it was just a matter of, no, and Nzinga is awesome. I'm seeing the comments coming through. Definitely a big thank you. Yeah, but the MCAT, super predictable exam, right? That last question that we did there, that is typical of the MCAT. I don't want to kind of, you know, milk it for what it's worth, but I'm going to milk it for what it's worth. I mean, this exam is just one of those things where you can really know what's coming as far as the test, and it's so predictable. And that's part of that MCAT expertise that is just so important for all of us to be fully engaged with and fully aware of, that it exists kind of as we're preparing for this thing and making it happen. So as I mentioned, this part here, and so a couple of reminders actually before I forget. Number one, be sure if you haven't already done so to enter yourself in the drawing, because we are going to give away a $50 gift card to Target so that you can buy all sorts of cool supplies to, or maybe just coffee to keep yourself awake when you're studying for this exam. So feel free, or make sure that if you haven't already done so, to fill out that link that's posted at the top of the chat. It's link number 2, so that's the one that you want to make sure that you're filling out. I see that session reminded me of everything I love thanks to Nzinga. Absolutely. So this is definitely good. Yeah, I saw a couple of you. I didn't want to steer away from the conversation, but yeah, book clubs are fun. So we do have a couple of book clubs around these parts. But always good time to get together with colleagues and friends and read something interesting and then discuss it. Speaking of things that are interesting, the MCAT, very interesting exam. So I'm just gonna move us over here to what we're gonna talk about. I saw in the chat a couple of you were asking already about this. So I'm just gonna kind of go through a couple things just to kind of give you a sense of how it is that you're gonna want to study. So when preparing for the MCAT, number one, you're going to want to give yourself roughly 3 to 5 months to prepare for this exam. Okay, so really important to give yourself the right amount of time from start to finish. So if you're planning on prepping or taking the test in April or May, now is like a perfect time to get started as far as your preparation is concerned. Number two, you're probably, and I saw a bunch of questions that came in that were asking about, how much time should I spend studying for the MCAT. Some of them we got privately, so that's important for us to talk about this. You're going to be investing about 300 to 350 total hours preparing for this exam. Now, you tell me, does that sound like a little bit of time, or does that sound like a lot of time? What do you think? What does that feel like? 300 total hours or 350 total hours. What do you think about that as far as that? And while you're answering that, I'll answer Rachel, I'll answer your question. When you take a class, generally speaking, you want the last class session, your last formal core session, to be about 3 to 4 weeks before the MCAT. Now the reason for that is there's going to be a lot that you're going to be doing in that time in between, right? So you're going to be taking full-length exams, you're going to be coming to the MCAT channel sessions, you're going to be reviewing material, et cetera, et cetera. So there's going to be a lot of activity that you're going to do. You'll have access to all your material up through test day, but just be mindful of that it is you want to give yourself a little bit of a buffer between the last day of class and the actual test. So I see most of you are saying that 300, 350 hours, that's a lot of time, right? That roughly equates to about 15 to 20 hours per week for about 20 weeks or so. So it's not, it's not a small chunk of change as far as your investment of time is concerned. But it's about how you structure that time. And I think that's, in the years I've been doing this, which has been a few, we've learned, and we continue to see this from students, that it's not just about how much time you put in, it's about how you structure that time. And so, what worked for your friend might not work for you. And I'll give you a concrete example. If you're a rock star at thermodynamics as a specific topic area, you're probably not going to spend that much time studying thermodynamics. So if your friend spent hours and hours studying thermodynamics, you probably don't need to do that because maybe she wasn't as good at thermodynamics. So it's a little bit about figuring out what's right for you and then structuring that time accordingly. Now, you don't want to study too much more than 350 hours because you will burn yourself out, and burnout is not a good thing. So what our program does, and what really the way that we advise students, and I'll advise you right now, in the early parts of your preparation, you want to be reviewing content and making sure that you are solid on that information. That is a huge part of doing really well on this exam, is making sure that your content knowledge and your content basis is absolutely strong. And we review that with you. I mean, we go through the high yield topics, we go through the lower yield topics, right? We go through everything. So it's, yes, points are points, exactly. So get the points from where you need to get the points from. It's a matter of collecting all the little bits of points along the way. So that's number one. Then, as you kind of get deeper and deeper into your studies, your review of the content is going to start to diminish, not because it disappears magically, it's because now you've become a master of that content and you need to practice the application of that content. But you don't want to dive feet first into taking full-length exams. What you need to be doing is you need to be doing smaller, kind of shorter quizzes, little exercises. And so we have that built for you. We have section tests, which just lets you take a section of the MCAT. We have something really, really awesome now. It's called our adaptive Q bank, where literally you pick and choose the topic areas that you want questions to be pulled from. We pull from a pool of questions, but the way the adaptive Q bank works is it's always giving you questions that are challenging to you. It literally adapts to your ability levels, and that is a unique feature that is only available at Kaplan. At the same time, you're going to be attending classes and you're going to be coming to your core sessions as well as your channel sessions. So the core sessions, everybody comes to all of those, but in the channel sessions, you go to the ones that are right for you. So you're going to be attending sessions that are what your individual needs are telling you that you need. And this is going to be live instruction, so you're going to be able to build a relationship, get to know your instructors, and really, really kind of build out what those individual needs are that you have in order to be improving your score. And these are all things that you can only get as part of our kind of complete preparation. And really, it's important to think about the proximal zone of learning. I mean, it's a super awesome term and something that we love and embrace. So the idea here is that you want to have structure, and that is what that's what we're here for. That's really the way that our programs are built, is to give you that structure, to give you that confidence. Now, there are lots of things that come with your program, right? There's practice, which includes practice questions. We got over 10,000 of them. This is actually a lie on this slide. We just updated. We now have 15 full-length exams. So it says 14, but we have 15 because you get all the available AMC materials. The AMC just added an additional full-length exam, so you now have 15 full-length exams through Kaplan. You have some of ours, some of theirs, or all, some of them are ours, some of them are theirs. So you get all of the stuff from the AMC. The books come to you. You don't have to worry about that. I don't know about you. I don't like carrying around books anymore. Those days are well behind us. So we have digital books. So you have printed books that you can carry around or leave at home. But we have digital books too. So you can literally have them, in the palm of your hand as you're working through. At the same time, it's important to be thinking about the live instruction that you have. And so we have the most live instruction available, period. I mean, you're not going to get more live teaching as part of your MCAT preparation anywhere else. Now, sometimes we need a little extra personal coaching. And it's like, if you go to the gym and you want a personal trainer, you can get one of those. For the MCAT, we have what we call our MCAT coaches, so our med school mentors. And what they do is they literally work with you for 3 hours, 3 one-on-one coaching hours, developing a personalized study plan that incorporates the feedback that you've gotten from the data, right, from the actual tests and the questions that you've taken, but then at the same time build out kind of the rest of your life and all the things that you're going to need to incorporate and really kind of act as a coach for you along the way. So that comes with our Plus program. So we have our core program, which is called MCAT Prep, and we have MCAT Prep Plus, which gives you the coaching hours, as well as our Foundations of Biochemistry, Psychology, and Sociology, for those of you who might not have taken those courses, because it's okay to not take those courses as long as you learn the material, and we will teach that material to you in those foundation courses. Now, I saw a lot of questions come through about the MCAT channel, so I'll explain it like this. Didn't mean to open up a can of worms. I'm going to open a can of worms up here. The MCAT channel is kind of like MCAT Netflix, except it's live. So you literally have the best teachers. It runs about 6 days a week. So we take Fridays off usually, but there's a rotating kind of series of 100 elective sessions that cover topics that are foundational to topics that are advanced and high yield. Really, the bottom line is you pick and choose the sessions that you need to go to so that you are, again, in that kind of key point as far as key areas as far as what your individual preparation is going to need. So here's what I want to do. I want to open it up to questions. This is a list of the stuff that we have available in our course. I mean, I won't read this to you. You can read it yourself. You can go to our website, find out more information. But I'm going to open up to you and see what questions are on your minds. Jordan, I'm glad you like the foundation courses. That's super awesome. Logan, you know, once a week, twice a week. It depends on your schedule, quite frankly. It's going to be a little bit different for everybody that's out there. So once a week live online. Yeah, so there you go. Gregory's taking it, but you're working. So good, good little balance that comes into play. So we have lots of options is what it comes down to. Now, a couple of you, I saw a few questions, and they're being bumped up to me here. We're asking about how the personalization works. So the way that Smart Reports is designed, and Smart Reports is the name that we have given to the system that prepares you for kind of figuring out what it is that you need to work on and what it is that you need to improve along the way. But the way the Smart Reporting system works is that literally, as you are taking tests, the Smart Reporting system is analyzing your individual strengths and weaknesses, and it is saying, here is what you should be doing next. So it's this adaptive learning technology. That's a very fancy way of saying is making predictions and recommendations of what you as an individual need to work on based on your previous performance. And the real cool thing about Smart Reports is that it only gets smarter about you as well as about everybody. So your information, as far not your personal information, but your performance information is fed into the system, and then your outcomes are then translated so that the next time you are working on something, it'll say, okay, when Petros did this and he needed improvement in this area, it seemed to work, and so now we're going to kind of take it from there and give you more recommendations to further build what your individual needs are. So the Smart Reporting area is, or Smart Reporting system is a phenomenal system, and something that you will grow to love. I mean, the nice thing about it is that you don't actually have to do the work to figure out what it is that you have to study. The Smart Reporting system actually guides you along the way through the process. Now, at the same time, and I said this to you earlier, and I'll say it again, we've got a lot of different options. I mean, we've got courses that are live online. I know Greg mentioned already that he's taking a live online course once a week, which is awesome. It's a nice balance for him. You've got live online courses, so that's literally live instruction. It's an interactive online classroom. Ask your questions, get them answered. You can take a course in person, so you go to a physical location. If you're more of a, I want to do this on my own, we have a self-paced program available, and then of course we have private tutoring. But what I said earlier was, if we want you to figure out what's right for you. So click on that link. I think it's number 1 in the chat roll that you can just click on that, and or it's pinned, I should say. So you can click on that and set up a time, talk to one of our instructors, and they'll help you figure out what's the right course that you individually are going to need. And so it's a big decision, but part of the decision, and this is where I want you to kind of be mindful of your surroundings. The Smart Reporting system. So Nicole, I see your question. Smart Reporting is a feature that's in all of our courses, right? All of our courses get Smart Reporting. So regardless of which of the 4 you choose that are on the screen here, you will have Smart Reporting. But what I was saying earlier a second ago was you want to be mindful of your surroundings. If you are planning on applying in 2018, so if you're planning on putting an application in during this upcoming application cycle, so that is an application in 2018 for entry in the fall of 2019, you are actually right now in the prime season. I'm not kidding you when I say this. You are in the prime season to begin your preparation. And the reason for that is ideally you want to take the tests by May or June so that your application is complete by June, so your application is being evaluated in the earlier parts of the admission cycle. Medical schools work on a rolling admissions process, so the earlier application is in, not that your chances are better, but they're going to be spending more time reviewing your application, and you're going to get a little bit of of an edge in the sense that your application will be seen by the admissions officers, not when there's this huge pile of applications that comes through their doors. Some med schools do report, though, that their classes fill up relatively quickly because they're not waiting around for students who apply late in the cycle. I don't want you to rush your MCAT. I don't want you to rush your application. I want you to do the application when the time is right for you. But now is the prime time to prepare so that you can be ready for either the April or May exams, or even the June or the July exams, just depending on what your kind of scenario is as far as your schedules are concerned. And to that end, we actually have a really nice promotion that's going on right now. You guys kind of get the best of the best here. If you enroll by January 31st, which is literally tomorrow, we will give you unlimited preparation for the remainder of 2018. So, normally you would prep for a May exam or a June exam, and your class would be linked to that, and that's when your expiration would happen. If you enroll before January 31st, you will have access to our materials through the entirety of the year. Now, you might be thinking, why do I need it for the whole year? Maybe you're not ready to start just yet, but you're going to be prepping in a couple of months. Get signed up, get the books, get access to the MCAT channel, start nice and slow, space it out. If things don't work out, maybe you need to kind of continue to study. You can continue to study through the end of 2018, and then if you still need to continue to study, then you can use your higher score guarantee, which allows you to repeat the course for free, whether, if you're not satisfied with your score, or if you weren't ready to take the test, or you get your money back if your score doesn't improve, right? So there's lots of options to you, but this is a really, really good time to get enrolled. We only do this once a year. I can guarantee you that this will not be extended beyond January 31st. We literally do this once a year. We do it at the beginning of the year to kind of kick off the MCAT year, and this is a prime time for you to get started on your preparation. Also, because you can save up to $300 off of your courses on top of the unlimited prep. We usually don't have double promotions, but right now you actually are getting there's a double promotion. So really, really kind of great time to get started as far as the prep. But as I said, there are a lot of different options, and there are a lot of different there are a lot of different pieces that come into play. So as we look at this, this is a little chart of kind of what comes with what. So all of our courses get pre-recorded lessons, the QBank, the AMC resources, the flashcard app, the smart reports, the expert instructors, and support from teachers. Our live options, so the live online or the in-person options, of course, also give you live lessons. So you have the live lessons that come through there. The MCAT channel is available as part of everything Self-Paced Plus and beyond. And then you can kind of see the rest of the chart here as far as what's included throughout the programs. But again, this is hard to navigate. I want you to ask your questions here. Let us answer them for you kind of as we go. That's what it comes down to. Nicole, when you enroll, you have access through your test date. So every class is linked to a test date, so you'll have access to that. So it's not for 3 months, but when you sign up in January, you actually have access through September of 2018, because that's the last test date that comes into play there. Logan, you had a great question as far as what's the main difference in the teaching style and the lessons for live online versus the self-paced package. Self-paced package is all pre-recorded lessons. Live online is literally live. I mean, you ask a question, you get it answered. And you don't just have one teacher that's on camera. You have one teacher who's on camera, but you have multiple teachers that are off camera. So it's actually more interactive, weirdly, because you actually don't have to wait to get your question answered. You just ask your question, and there's somebody who's going to be able to, who's going to be able to answer your question accordingly. So it's super awesome. John, the MCAT prep package does include smart reports, absolutely. The one that's the comprehensive courses. So MCAT prep or MCAT prep plus, those include and as well as self-paced or self-paced plus. Those do include smart reports for sure. A couple of you were asking about affordability. We do have tuition assistance. So if you apply for tuition assistance and qualify, you can save up. You can basically get 60% off, up to 60% off your course. So that is based purely on your estimated family contribution, which is a number that you get from your undergraduate financial aid office, and then and then your GPA. So those are the two bits. So if you want to find out about tuition assistance, you can literally maybe we can put the link up, but it's just Google Kaplan tuition assistance, and it will pop up. So that's what it comes down to. Logan, you need to sign up before midnight Eastern time for the unlimited preparation, and then the up to $300 off. So that literally ends at midnight tomorrow, the 31st. So end of the month is when that's coming through. A couple of you might be interested in something a little bit different, so I figured I would speak to it for a second here. We have, one of the beauties of Kaplan is the convenience of our schedules. I mean, you will find a schedule that will work for you. We have so many class options that are available, but sometimes you might want something that's just a little bit more intensive, different from tutoring, different from a class. And this is actually our intensive programs. Our intensive programs are 5 or 6 week, for lack of a better term, boot camps that you either can do live online or in person. We have one that's live online that's coming up in the spring. We have some in the summer. We have 3 in the summer, Atlanta, Boulder, and San Diego that are coming up in the summertime. Those are in person. These are, full disclosure, you fully just work on MCAT. It is a very, very structured setting. It is, you're almost in, I don't want to say isolation, but you don't want to be doing a whole lot else during this period of time. So it is a very, very intensive period of time where you can concentrate and focus. We have our top faculty that work on the intensive programs. We put you into small groups. So you have your small group presentations, you have your kind of group lectures, and at the same time you have private tutoring. So it's a very, very intense program. And if you want to find out more about that, click on that link at the top of the screen there, and we can get you set up with somebody who will tell you all about that. So I think it's time. So Shwarya has notified me of our winner. Are you guys ready to find out who the winner of our $50 gift card is? Keep your fingers crossed. So we'll take a drum roll. Drum roll, please. The winner is Mark Sandifer. So Mark Sandifer, congratulations. You have won a $50 gift card to Target, which you can use on anything that you can find at Target. We'll be in contact with you about your prize. So congratulations again. But we will stick around to answer any and all questions that you have about what your study timeline should look like, what you need to be doing, what you shouldn't be doing, questions about our programs, and figuring out when's the right time to get started and all those things. So we'll stick around and get those questions answered for you. So congratulations again, Mark. Sorry to all of you who didn't win, but you can come back tomorrow because we're going to be doing MCAT Bootcamp tomorrow, and I'll actually be hosting and I'll be teaching. So I'll be doing both of those. I'll be looking forward to it. So MCAT Bootcamp is one of my favorites, and really looking forward to seeing you all as far as that event is concerned. And we'll have another prize tomorrow. So that's that's even better, the better part of all of it. So let us know what questions you have, and we will get those answered for you here. But otherwise, thanks again for joining us tonight. We will, if you have questions about any of our programs, again, please feel free to click on that link or reach out to us, give us a call, 1-800-CAP-TEST, and we'll be more than happy to kind of walk you through and figure out what the right thing is for you. I hope our paths cross again. Yes, I'll see you guys tomorrow, and I hope our paths cross again. Maybe they'll cross again in the MCAT channel. Who knows? So I have literally run into students on the street who've said, "Wait a minute, I saw you teaching the MCAT the other night." So it is one of those things. But yes, happy setting. Big thank you again to Nzinga and to Shwarya and to Patrick and to Timothy. And there's a long list. Carrie and Brandon for being here tonight. Big thank you to Greg. Greg's been running things behind the scenes very well. So big thank you to everybody, and big thank you to all of you. Big thank you to you for joining us. And as I said, let us know what questions we can answer for you. That's what we're here for the next 10, 15 minutes or so. As questions come in, we'll be more than happy to get those answered for you. But have a good night, everybody. Take care. And as I said, if you have questions, just feel free to pop them into the chat. We'll get them answered for you along the way. And if there's anything specifically that you want me to go over, more than happy to go over it as far as MCAT and prep is concerned. John, I'm glad you're inspired. I'm glad you're inspired. It's good. Glad your brain is working again. Cool. All right. Well, if you have any final questions, let us know in the chat. I'm gonna hop off camera, but everybody have a great rest of your evening. And because I don't see any questions that have come through, so I'm guessing we answered all of them for you. So nicely done. Good work, everybody. Have a good night.